Welcome to ModPath Chat, the official podcast of modern pathology, featuring interviews with authors and experts on the latest science, technology, and developments in the field of pathology. Our host, Dr. George Netto, is the editor-in-chief of Modern Pathology and professor and chair of pathology and laboratory medicine at Perelman School of Medicine at the University of Pennsylvania. Here's Dr. Netto.
Welcome to ModPath Chat. My guest today is Dr. Arnaud de la Fouchardiere from the Department of Biopathology at the Centre Lyon Bernard at the Universite Claude Bernard in Lyon, France. I said it in one sentence. Arnaud is an international expert in melanoma. His research focus is in the molecular classifications of melanocytic tumors. He is a regular contributor to the WHO Blue Book on Skin Tumors. We will be discussing his teams. It's a multi-institutional team that include many important collaborators from University of California and San Francisco. Together, they put a great modern pathology study on melanocytic neoplasms with protein kinase C gene fusions. Thank you, Arnaud, for accepting my invitation.
Yeah, thank you. Thank you, George, for having me today. And hello to everyone.
So, would like to start by a little background. Not all of us are smart melanocytic or dermatopathologists. So give us a little background why this is important and what's new about this.
So there's been in the recent years a lot of discussion about how to classify melanocytic tumors. And there's slowly the emergence that there can be like several diseases that can be grouped or subgrouped according to molecular data. So there's been a lot of unknown cases, but the growing knowledge with the new technologies and with studying more samples is starting to give us more insight on precision medicine with new markers, new biomarkers as genes that will help us to make this big group. So there's been a start in the fourth edition of the WHO that started listing a number of genes. And but the story is ongoing as we're discovering new gene fusions, new mutations, and new combinations of things. So it's slowly shifting and moving towards a more robust. So I think this kind of discovery is part because it's going to change where one group of lesions is going to move from one part of the classification, which was the common nevus, to the part of the blue tumors, like blue nevus type of lesions. And these are kind of like two diseases from the genetic point of view.
Wow. So as long as it doesn't become like neuropathology where it's all molecular, we're okay with that. For the poor man like me, we still need a little morphology, but it's amazing. So, and these classifications matters. I take it from term of biologic, not just based molecularly. It matters biologic behaviors and hopefully targets of therapy. Is that?
Yes, but as a pathologist, what is really fascinating is that there are very strong correlations with the morphology. So it's not just sending an unclassified tumor. Then, if you start to know about the morphology's specific features, you can kind of guess what is going to be the genetics. And this is the whole interest as a pathologist, to invest in this correlation between the molecular features and what is the genetics behind it.
Awesome. So let's dig in. Tell me a little bit about the cohort, where the case is, and what, just briefly, what techniques you applied, and then we'll talk about the findings.
Yeah, so this work started at the World Melanoma Conference in Australia, in Brisbane, and we worked with EYA putting together. We noticed that we had an overlap in this new genetics, which were PKC fusions. And so we started collaborating from there. So this story started in 2017. So it's already quite a few years. And the cohort is even old because it goes from 17 to 19. So we have all the experience in that, and we have more like double of the cases. So we started finding that we had lesions that were having the same genetic anomaly, but they were different grade. Some were benign, some were malignant. And what the most interesting part is they had a very various morphology. So it was, it was a field that I found so confusing. This is why I studied these lesions, because I could not classify them. So I put them in whole RNA sequencing, which found these PKC gene fusions.
Right. So it's protein kinase C or PRKC, and we'll talk about the findings in a second. So I guess to summarize, so some of them look to you benign and they have various morphology, but when you apply the RNA-seq, you start finding commonalities in fusions, and wow, fascinating. So yeah, so I think it's the total is 51 cases, right? I gather most of them are from your consultation.
Yeah.
Very heavy consultation.
Yeah, a lot. Most of them are from the consultation. In the initial concept, as if you look at the literature on PKC, their only description is hyperpigmented lesions that had been grouped with the PEM, pigmented epithelioid group. So here we're bringing new morphology with unpigmented lesions, so that's really an. There are some features that are very common to the blue group. So it's like transitioning from one group to the other, and changing the name, which is something very difficult when you find something new, is how to call it correctly. But it's always a work in progress, and you're always judged by the future about how you did it well in the first place.
Correct. And so briefly, can you tell us how they look? You have some beautiful images in the paper, and definitely the audience need to look at them. But can you just, how many, groups, you mentioned different looks to it. So can you share with us?
So that's really the main difficulty is explaining to people, but I'm going to try to do it, is right. That it can be broken down into several features, and that I will list. So the first one is like the PEM-like is the typical one. It has hyperplasia of the epidermis and hyperpigmentation, which is not at all the hallmark of blue tumors as we have learned them in med school, which we always thought is like, it's only dermal, it's not supposed to have a junctional component. So here is novelty that we bring with this entity as potentially an important junctional component to the blue group. And then we have the dermal aspect, which is something that has been described. When you find something new, the odds is somebody already described it, and it's very interesting to go to literature and find how people have called them in the past before having the genetic data. So doing that and looking at a lot of textbooks, the images typical of these tumors are often called combined blue nevus. So the second, this is the predominant aspect. It will have like a biphasic architecture, meaning part of the architecture is small nests of nevoid melanocytes, which were in the combined, which means it has two different morphology, were called similar to common nevus. And the second part is, of course, similar to blue nevus and has dendritic, hyperpigmented, and spindled cells in a fibrotic background. So this is like probably the most common finding we have in these tumors. And also it can be like a horizontal band that is in the upper dermis of only nevocytoid cytology. So it's going to be a combination of all this, and you can have all the variations. We can have only one feature, all of the features, or a part of the features. So this is where this is why probably it took years. And I remember a few years ago, I gave up trying to classify these tumors before having genetics, because I could not make sense of this diversity in morphology. But now we're starting to have the tools to work on this. So what is also interesting is that there's a lot of small features in blue nevus, like it can have multinucleated cells. It can have hyperfibrosis as a sclerosing type of blue nevus that we know is frequent on the scalp area. It can have a foamy cytoplasm, kind of clear cell variant. And we find all these variants in the PKC fused, which also makes sense as they are part of the blue group.
Great. So I guess if I'm looking at a lesion, and by the way, what age group? Because I realize some of them are in young patients, right?
Yeah. So the age group is very wide, but so there are some very young patients. There are congenital lesions. There are giant congenital nevi with this anomaly. And there are even some, most of the melanomas were in children. So we have children who died from this type of tumor as a malignant evolution from a giant congenital nevus. So that means there is a potential to become malignant that is important to know in such tumors. And usually we found a few atypical intermediate group lesions and some malignant ones. And most of the time you can see in the background the biphasic benign tumor, meaning this is a clonal evolution from a pre-existing lesion.
And just to address that, the evolution is also molecular evolution, right? In terms of via the BAP1 inactivation.
Yes. So we know like that blue melanomas are very similar to uveal melanomas, and in this group, the bad outcome is often related to the inactivation of BAP1 gene. And here again, we have found this BAP1 loss that can be studied by immunohistochemistry very easily. It's most often the immunochemistry is the best tool to analyze this loss. And here again, it has a feature that these are the ones who might have the worst outcome. So that is, we find the secondary events that also vouch for putting this tumor group, this PKC fused, in the blue group of tumors.
So it sounds like at least you'll have some morphologic features that may point to malignancy or borderline, and if you can use BAP1 to help you prognosticate, I guess, and know that these have potential to metastasize and what have you. Is that because I'm trying to think how practically we just have to send every lesion to RNA-seq, or how would you approach it if I was facing something like that?
Okay, so in fact, when I show this at like a local staff to a lot of people have already seen these tumors. These are quite common, so it's not very a rare tumor. But it's mostly like it looks benign, so people just call it like a blue and just move to the next case. But so they'll be able, with maybe looking at the figures from the paper, to recognize something they've seen that was strange. And these are benign tumors, so they just should be treated like a blue nevus, like a complete resection with narrow margins is sufficient. And so in the progression, usually you will see mitotic activity and Ki67. That's already very easy tools to be worried. And it's like the disappearance of the normal biphasic architecture. So, and also I don't think that they should be studied with RNA-seq anymore. So we put up a FISH test for this, and it's based on the frequency. So out of the 51 tumors, about 2/3 of them had PRKCA fusion. So that was the predominant feature. And then 1/3 had PRKCB fusion, and only one case had PRKCG. And these ABG are part of the same biological subgroup of PKC, which is a very wide group. And so currently, for confirming these lesions, we will do first PRKCA FISH, And if it's negative, PRKCB. And if we still think it's that, we will go to RNA-seq to study, maybe discover a new anomaly. And that's how we found some new stuff also.
And probably that's more pertinent if you're worried about it being malignant, you're seeing the features that you pointed, because then you want to categorize it as such and do BAP in the same time, right?
So for the malignant part, I think array CGH is probably the best approach to assess the progression. So we clearly saw that the benign had no anomaly by CGH, intermediate had a few anomaly, and there were major anomalies in the malignant ones. So there is really, it's going to be the major test to help you distinguish the three groups.
Amazing. It's getting very, very complicated. And but at least it's good to have awareness of this in a blue nevus in a young patient, that you have a reason to worry about, and it's not fitting exactly what you usually encounter. And so what is the next steps? I think it's like everything else where you start defining the molecular underpinning of a group, bringing group together, you'll find even more variations as people start doing FISH and that. And does it really have any treatment implications or any management implications, being this category of fusion versus others?
So that's we've seen that they can go to really aggressive mode, especially in young children. And I know for uveal melanomas, there have been PKC targeting specific therapies that have been tried in recent years. But I don't know if they have been tried in this type of anomalies yet. So that's another question, is that since we found them in skin tumors, maybe they will also be found in uveal melanomas, and maybe also in leptomeningeal melanomas who have similar genetic backgrounds and could maybe benefit from these specific PKC targeting therapies. This has to be assessed, yes.
Amazing. Well, thank you very much, Arnaud. Any thing else you want to say about this topic? It's really, I definitely enjoyed the conversation.
So I really I've seen maybe 100 tumors now, and I'm trying to put together a video that will show all these 100 tumors on my YouTube channel. So people will have the opportunity to evaluate the diversity of the morphology in these tumors, and hopefully it will help them recognize them, because I think they're quite frequent and most pathologists will encounter them.
Wow, good. You're already competing with our podcast, but that's fine. I will let you do that for melanomas. That's wonderful. The more education, the better. So definitely our audience will hopefully go to your YouTube and learn more about this. Thank you very much.
Yeah.
Any opinions expressed in this podcast are the speaker's own and do not represent the views of Modern Pathology, Elsevier, the University of Pennsylvania, or USCAP. Your ModPath Chat host and scientific director is Dr. George Netto. The producers are Dr. Catherine Ketchum and Carly Hess. Technical direction is provided by Kaminsky Productions. Music by Mitch Neubauer. Thanks to the authors, reviewers, and editors of Modern Pathology for making this podcast possible.